We have this great presentation by Guillaume, but I first wanted to thank Fars and Vafa for putting together so many nice seminars during this year. So thank you.
I can say thank you for also helping with put all of this together. Next event for Common Sports will be last one, but we'll be back in the fall. We're taking the break during the summer. We're delighted to have Guillaume Duclos from Brandeis. So we're talking about building active nematics and active liquids topological machines. And as usual, we encourage questions. So if there's any questions, just feel free to interrupt and ask. If you're in the Zoom audience, you can either just unmute yourself and ask, or you can just type in the chat, and I'll be monitoring it so that it's whatever you guys prefer. So cool. Yeah, let's get started. Thank you.
All right. Thank you for that. Thank you, David, for the invitation. So yeah, my name is Guillaume Duclos. I'm an assistant professor at Brandeis University, 10 miles from here. And I've been there for like 3 years. And what I'm going to talk about is some of our like kind of recent progress towards like trying to build active materials out of cytoskeletal proteins. And so this talk is really like in two parts. So I'm going to first kind of thank the students and postdocs that were involved. So that's us on the Brandeis campus a few weeks ago. So the first part is going to be work led by my graduate student Najma, here in collaboration with like Brandeis and our student Minu, where we worked on materials that are composed of microtubules and kinesin motors. And the second part of this talk will be about actin-based materials. And work done by my postdoc Joe Lopes in collaboration with Thomas Fay in maths and Bruce Good in biology, both at Brandeis. All right, so my lab is, and so we run an experimental lab, and we're interested in the emergence of collective behaviors in living systems. And you know what's really striking about this movie is that, you know, like whether we're talking about sheep, fish, bird, you see very generic patterns that emerge. And so it doesn't depend on the type of particles that you're like looking at. And so because we want to investigate, you know, what's the physical mechanism behind the emergence of these collective behaviors, and because I cannot fit 10,000 ships in my lab at Brandeis, we turn into biology and we look at smaller systems. So we look down our microscopes and take a look at cells, bacteria, tissues, embryos. So those are kind of different examples here that show how like rich living materials, living matter can be for like physicists to kind of study the emergence of those collective behaviors. So, you know, like those three movies are like different time scales, different length scales involved, but really the mechanisms that drive them out of equilibrium is always the same. You have motors and polymers that are out of equilibrium that consume energy at the micro scale, and that's what drives at the larger length scale the emergence of these types of patterns. All right, so really what we want to be doing is not really study sheep per se, but to look at what are the symmetries that are conserved, what's the ones that are broken, and try to identify generic physical mechanisms that explain the self-organization in these systems. All right, so basically, whether we're looking at a sheep or like a bacteria, you could think of it as a self-propelled particle that has a head and a tail, and they're going to assemble into this very nice like ordered phase that is active. Like each particle is moving, and so as a really like a striking example of like how very different systems can self-organize in very similar patterns. Here I'm showing like two examples. On the right, the same kind of flock of sheep. On the left, that's an experiment from the Bausch lab in Munich, where you have micron-sized polymers that are moving, that are crawling basically on a glass slide that's covered with myosin motors. So the myosin motor is basically are pushing along, sliding along those filaments in 2D, and they bump into each other. The line, you see the emergence of those beautiful kind of polar vortices.
And the motors are anchored to the cover slip?
Yes, here's an anchored to the cover slip. All right, so some of the research goals in my lab. First one is to identify generic emergent behaviors in active systems. 2, we try to study some of the fundamental design principles that underlie biological function, for example, related to cell migration and collective cell migration. And finally, we want to try to use this knowledge to create new materials, active materials that behave like cells do, but much simpler methods. And so now we want to look at biology, look down a microscope and try to get inspired by how cells move in order to replicate that in vitro in a test tube. And so this way of kind of doing science, getting inspired by like living system to try to design something like a new machine is not a new way of doing science. Da Vinci spent like the years kind of looking at the optimal shape of the wing of bats, the nature of air to try to design a flying machine. Back then, that was like a crazy idea, you know, like, but we finally did it after a few centuries. So can we do the same thing looking down at living cells? Can we look at what are the different components that are inside the cell, identify the ones that are relevant for certain function, let's say for motility, for cell division. We purify those proteins, reassemble them in vitro to try to recreate a simpler model of a, like kind of a simple cell, let's say, right? And so, you know, like basically what we and others have been doing is to purify some of the components, some of the proteins that you find inside the cell. Right, so you can think of them as like kind of building blocks that are like active. We try to, you know, like use what we know about biology to identify proteins that might be relevant to create active materials. So motors and other types of proteins that can generate forces inside the cell. And then we reassemble them, put them in a test tube and see what comes out of it. Okay, that's and then after that, after the fact, after we've seen the type of emergent pattern that arise, we try to rationalize them using like symmetry arguments. What I think we should really think about in this field of like, you know, like experimental active matter is, can we do the reverse? Right? Can we try to rationally design active materials that have a targeted state, targeted self-organized state? So basically, if I want to build this, what proteins should I use? In what amount should I use them? You know, like how should they interact with each other? And so that's actually very complicated to do that because even though we know a lot about the isolated building blocks, right? There have been a lot of biochemistry studies, for example, on molecular motors, on the stored force, on like kind of really what's happening at the micron scale. We don't know how to go. So this is a multi-scale problem. We don't know how to go from kind of the energy injected at the microscopic scale to the emergence of these patterns. And that's a challenge both like experimentally, but also numerically, right? So we don't know how to coarse-grain particle-based simulation to try to extract hydrodynamic parameters, for example. And so what I'm going to try to do in this, in the first part of the talk is try to see if we cannot do the reverse problem. So try to rationally design these materials. Right, so my talk is divided into 2 parts. First, I'm going to talk about instabilities in active nematics that are composed of microtubules and molecular motors. And then in the last 15 minutes, I'm going to talk about some new experiments where we try to get rid of the molecular motors and instead use polymerization and depolymerization forces as a driving force for these types of activities. Okay, so the materials I'm talking about are basically composed of cytoskeletal proteins. In particular, we're dealing with like filamentous polymers such as actin and microtubules that you find here in this cell, where like both are like labeled in different colors. What's possible is to purify those proteins, so isolate them from the cell and reconstitute them in vitro in the test tube, basically.
So is there a nucleus in the center in the lower left somewhere?
Hard to say. There might be a nucleus. It's possible to remove the nucleus too sometimes. So
Okay.
here, because it's like just what's fluorescently labeled, you see just what's fluorescently labeled. So the nucleus is not labeled, so you don't see it.
But if it were there, would the microtubules be piercing it?
No, they would be like
They would go around. And then vimentin is too floppy so that people don't use it in vitro as much.
So okay, so it's been much less used in vitro. It's too floppy, and like I think people haven't really identified like what's really how it works, what it does. You know, like like what's the role of vimentin. People have seen that you can knock it down and it impacts how cells move and their shape, but really we know much less about vimentin than what we know about actin and microtubules. So I think that's why, in terms of like in vitro reconstruction, people have been focused on those two things.
And are there motors that do walk along intermediate filaments, like microtubules?
Not that I know of, but maybe someone did understand. So that's another reason why most of those like active, like reconstituted materials have been based on molecular motors and cytoskeletal filaments. And so we know a lot about different types of motors on microtubules, same thing for different types of myosin on actin. And so just been easier to kind of purify this.
Thank you.
Yes.
You know whose movie that is?
I don't remember. It's online. Very online. It's very pretty. All right, so you can purify. Do you know who it is?
No,
Okay.
I love the didactic question.
So it's possible to purify those filaments and rebuild the microtubules in vitro. So here's the microtubule that those like cyan rods here. They've been they're immobilized on a glass slide, and those tiny dots that you see are the kinesin motors. So molecular motors that basically can hydrolyze ATP, and they walk towards the plus end of the microtubule. In cells, they're responsible for cargo transport. So you find a lot of them in neurons, for example. So what we do every 3 months, we basically purify those this protein out of brains, and then that's how we can get a lot of material that we use on a day-to-day basis. Okay, so
Are they bovine brains?
yeah, bovine brains. Every time the kinesin hydrolyzes a molecule of ATP, it takes an 8 nanometer step onto the microtubule. And so, you know, you can reassemble these types of material in vitro. So like we have microtubules, clusters of molecular motors, and this microtubule cross-linker that you find in a spindle. You can this way create a network out of the system. So that was work that was pioneered by Zvonimir Dojic in early 2010s at Brandeis. And that's a movie from his group here. In the first paper where they studied these types of active materials, whereas here they kind of focus on just 2 microtubules that randomly come together, and then you see that as soon as they come, they're going to slide apart. Right? So the sliding is because they basically, what you don't see here, you have a lot of those molecular motors that are basically stepping on both microtubules and induce the extension. Right? This is immersed in liquid. So what's happening is that as this bundle extend, it exerts a diperoxidant side force. So you're pulling fluid along the short axis of the bundle and you're pushing along the long axis.
They found a way to label the plus and minus ends.
No, here it's just manually.
Just manual.
Yes, so it's just after the fact because they know that they have to be anti-parallel to kind of.
But then if you switch them, you get the same dynamic. So it must be
Yes,
guessing.
it's just about the parallel versus
Yeah.
anti-parallel. And so you actually see at the end that they come parallel and then they don't slide.
Yes.
So they only slide when they come anti-parallel. All right, so that's a movie where it's very dilute, but if you start to increase the concentration, you get this percolating network of microtubule bundles. That's still like quite dilute, but now you see those bundles are continuously extending, buckling, and reforming somewhere else. And so this is like, I say it's still dilute, so you're not in a nematic phase, you're below the ion transition. You don't have any birefringence here, only if you look very closely, like inside the bundles, because that's where the microtubules are tightly packed together. But between 2 bundles, right, you can see in this like 3D confocal movie, you have a lot of empty spaces, it's just liquid. And what I want to also like point out here is that this is a 3-dimensional system. Right? It has a certain thickness. It's pretty small. It's only 100 microns here. Right, so much so smaller than like this K bar, but it still exists. What's quite striking though is that like the length scale of this flow, right? The size of those vortex is like a few hundred microns. So it's orders of magnitude larger than the step size, 8 nanometers, and it's larger too than the length of the microtubules that are only like a 1 or 2 microns. Right, so the question is, how do you kind of go from kind of nanometer step size to this like large scale micron size flows? And so to investigate that, what we're doing is that instead of looking at this kind of chaotically moving active fluid, we flow align it into a microfluidic channel. So we take the same disordered material, and because we flow it in a very thin sample, thin channel, the shear basically aligns the microtubule. So you shear align the microtubules, and now you're like have this initial very long range nematic order, even though you're still below the ion transition.
So before you go in this fascinating direction, just a brief question about the previous old experiments. They're churning along there with the ATP being burnt up.
Yes.
And some people vaguely make an analogy with turbulence and so
Yes,
forth.
yes.
Is there a cascade in this system, a recognizable cascade?
It's
if you look
hard
at the
to-
velocity field and stuff?
It's hard to say. Some people are looking at this in the 2D active nematic that's much denser, and they say that you have some cascades, some scaling, but
It's been done with bacteria, for example.
it's been done with bacteria, and recently there was like a paper that was a 2D active nematic, but it's like, yeah, it's still, I think, still like an open question.
Okay, thank you.
Yes.
Just experimental detail, but you said it's bundler, these older experiments?
So yeah, in the older experiments, they were bundled with PEG,
Okay, yeah.
which is a non-specific depletion. Now we're bundling with
Now you
this crosslinker, and it gives very similar.
It is very similar in every aspect.
In every aspect, what's great now is that because it's very the crosslinker is specific to microtubules, we can create composite materials. So we can mix this with a lot of other types of soft materials and try to like activate.
Okay, but other than that, the PEG does the job.
Yeah, yeah.
And the crosslinks are reversible.
So it's it's like it's crossing. It's a transient crossing. It's going to be important actually for the rest of the topic. Anyway, so we start here with this like well-aligned states, and then we stop the flows. The microtubules now stop moving, and what you're going to see next is only flows that are driven by the local extension of the bundles. Okay, so you see basically this very well-defined stability that has a finite wavelength. The length scale here is like a few hundred micron. There's a couple of features here that I want to point out. One is that you have a finite length scale. Two is that this instability is in the plane of the microscope, right? So it's still a three-dimensional material, thickness of 100 microns. The width is probably like twice the size of the screen.
So it's just an illusion that my eyes see ridge lines coming out of the plane.
Yeah, it's not coming out of the plane here.
Yeah.
It's really everything is in plane.
And eventually it reaches, you tell us this, I guess, does it reach the statistically steady state?
Yes, so what you see at the end here, you start to see like a second instability of it.
Yes,
So you have a cascades of instability. Each one you have noise that accumulates, and at some point you just lose like all memory of the initial like. So you have cascades of instabilities here. What's tricky is that you see the bundles, they coarsen a little bit, so the wavelength is also like changing, cycle is increasing, but eventually you reach a steady state that I was actually.
So you initially shear align, right? So how does the distance between microtubules compare to what you get in the bundle? Like average distance?
Yeah, I don't know. It's hard to say. I think actually, Zvonimir and Dan have been trying to like measure this thing, but it's quite hard to do. But definitely by just looking at the structure of the bundles, it's coarsened over time here. Like at the beginning, so the shear kind of like breaks up the bundle.
And I'm sorry, you activate this, you trigger it by adding ATP or something?
No, there is already ATP. Everything is just kind of really like a very strong shear flow aligns everything.
I see.
Right? So actually we've done experiments too when from this state you kind of trying to apply an external shear, and if it's like if the shear is strong enough, you can as well kind of like realign everything.
And the shear is just between two flat plates?
Yes, exactly.
And that determines the direction.
Yeah, so that determines the initial like alignment.
Yes, thank you. When you stop the shear, this happens.
Exactly.
Sorry, one quick question, so you put it in a tube-like structure?
It's no, it's a like kind of channel like this. So it's very thin, very long and wide.
I see, and the longitudinal direction in the next movie is the x direction,
Yes, that's this direction.
and the out of plane direction is the height.
Yeah, that's it. Exactly.
I see. And does the wavelength depend on the height?
Yes, yes, I'm going to talk about that. That's actually what matters is a height of the
And
channel.
it's thin enough that any thermal effects are irrelevant.
Thinner than
Any thermal gradients are irrelevant.
yeah, yeah. I mean, it's at room temperature.
But it's also thin so that that you can
Yeah, it's,
get good,
yeah. It's,
you don't have Rayleigh-Bernard instability.
no. Actually, you know, so that's would be the ground state, right? If there was like no activity, like something that's free, right? So if you remove ATP, you remove the motors, you just like stay like this for like 12 hours. So it's really like activity driven.
Is this with the pyruvate-based system?
Yes. So there's an ATP regeneration system that allows to kind of keep the ATP at a constant level. Okay, so let me try to explain what's happening. Why do we have this instability? So imagine you have swimmers like these pusher swimmers that exert a dipolar extensile flow. And I'm going to start with a suspension that is aligned like this. If I impose any small bend distortion like this, infinitely small, and I have an infinite system, if I look locally, you know, like the extensile flow gives rise to a shear, and so now you have elongated rods in a shear flow. That's why they're gonna realign with the shear. So basically, any small bend fluctuation we get enhanced and like grow over time due to the extensile nature of the microtubule bundles.
Sorry, before we go, there's a question from the Zoom. I
Yes.
think I missed it. So it's from Kyung-Soo Kim, and the question is the flow is coming from left to right. I think this is the previous.
So here,
Yeah.
so the bundles extend these
I'm not
volumes
sure.
along axis, and that creates flow perpendicular this way. So like along inside the plane again of
It's up and down.
up and down. Right? Okay, so the conclusion here, and that's been known like for a while from like theoretical studies, that like the bend modes are unstable in extensile liquid crystals. And so this instability can be described by, you know, the linearized equation of Navier-Stokes for like a 3D active nematics that's extensile. And that was done, there was like work done theoretically already 20 years ago by Simha and Ramaswamy. And what they've seen is that, you know, like, and so we use the same equation. What we see as well is that basically the bend modes are unstable. So you see only pure bend deformation, and the wavelength is infinite. Right, so here the fastest growing wavelength is the wave number 0. Okay. Now, what we've done is to introduce no slip boundaries, and that's changing several features in this instability. Right, because in particular now there's only like a finite range of wavelengths that are unstable, and that's because the presence of no slip boundaries basically constrain your velocity field and your orientation field. So you cannot have pure bend deformation anymore. And so
So you say no slip, this is at the top and bottom of the parallel plates.
everywhere, so like in 3D, except that those are closer together.
And if you put grooves on the plates, then you can suppress this instability.
If you put grooves on the plate.
It's a standard way of aligning pneumatics, for example.
So okay, so that doesn't work here because it's a colloidal liquid crystal. So that works well with like those like kind of oil-based like semi-tropic liquid crystals, but here
It's not easy.
not really. What we should try to do instead is to put motors, I think, because now combining basically the motility assay with this, and it would be able to like flow more. So yeah.
And there's no, to go back to the early work, there's no splay instability, but that's
No,
stable.
exactly. So your original work here, it's on here by instability. Now here, we actually see two instabilities. A bend twist instability and a splay twist instability. But if you start from like a well-aligned system, the bend twist grows faster than the surface. Yeah, in the theory.
Have you looked at what is the boundary condition for alignment at the top and bottom?
So it doesn't matter. It's only like what we do is only a condition on the floor, so no flow condition. But we did look and we see that you don't have like a uniform profile of the orientation. So you're more bent at the center of the channel that you are on the edge. You still have a little bit of like distortion on the wall, so you're not anchored. But so it's the simplest basically boundary condition we could think about was just enough.
But for the nematic, it's like a weak anchoring condition.
There's no so experimentally or like in one? Experimentally, there's no anchoring. So we treat like the channel so that there is nothing sticking. So here basically is a short wavelength mode, have a larger fraction of band compared to plane twist. And so that's why the long wavelength modes here are basically inhibited, because it costs too much. There's not enough kind of activity to kind of overcome the elastic cost of those two distortions. Now in the small wavelengths, right? So wave number 1, you recover the same effect as a bulk, whereas the nematic elasticity basically prevents you from going wavelengths that are too small.
But the preferred wave number is what are the gap between the plates, the preferred wavelength?
Yeah, it's a little bit smaller. Yeah, it's in the same order.
Same order, right,
Same order,
the gap.
yeah, yeah. But it's not just a gap. So it's a non-trivial wavelength. It's not just a
Absolutely,
noise gap.
but it often does come out. to be that.
Yeah, so actually what I'm not showing that here, but like we did experiments where we changed the width and the height, and what's important is really like the height of the sample. So like the smallest dimension in the system basically sets the wavelength, because the smallest dimension basically sets the larger vortex you can create in this situation. And so that's quite striking, and like basically the wavelength of the in-plane stability depends on the out-of-plane confinement. The second thing that's important is that the direction of the instability also is perpendicular to the stronger confinement. And that's because confinement basically suppresses the fluctuations. So the fluctuations grow faster along the y-axis because the walls are further away from each other compared to the z-axis. And so in theory, you have a superposition of like all those modes, but like because in experiments we just see the one that grows the fastest, it's the one that's like.
But eventually you do get instability in the.
So we don't see that here. We only see the one that's in plane. But like I'll talk in a minute about another type of instability, which is this time out of it. Okay, so after the instability, like work done by Dogic in 2D showed that like when you have the growth of this distortion, you start to nucleate topological defects. Right. So basically, because you have a very large surface area here where you have distortions that are increased, at some point become energetically favorable to start to nucleate singularities that will concentrate all the distortion and relax here in the center the system, right? The strain here. And so you basically have this plus half defects here that's migrating away from this minus half. Okay. And so since that work in 2012, there've been a lot of studies, and I've seen that you guys have like a few talks that talk about that already, about topological defects in two-dimensional active nematics, whether it's microtubules, bacteria, or like epithelial cells. And what's been really like great to see is that the generic features are driven. So the generic features are conserved in those systems, right? You have motile plus half defect that basically drive the chaotic dynamics. But what I want to talk about next is what's happening now if you're in 3D, right? Once you're in 3D, you don't have just bend and splay. Now you can also be able to twist in a third dimension. And that will really change the type of defects that you will be able to see. So because we're talking now about a 3-dimensional material, we need to use 3-dimensional 3D microscopy. So now I'm going to present work I've done when I was a postdoc with Zvonimir. And in collaboration with Sebastian Stration at UCSB. So Sebastian, the physicist-based studying embryos, purified embryo. And this microscope that he built here, the Light microscope, allows to kind of look very fast at 3D volumes that are extremely large and do that with an isotropic resolution because you're able to rotate your sample so you can look at it from different views. So that's an amazing microscope, and we basically leverage it to kind of study these three-dimensional active nematics.
Does it scan or raster in 2 dimensions as well as going to the third?
So yeah, so it's you have a sheet basically, right? That scan your sample.
Aha.
And because you can also rotate your sample. So okay, so the issue when you do any types of 3D microscopy is you have poor like resolution on the z-axis.
Yes.
Micron to microns. But now because you can rotate your sample, you can have one view that has a good resolution into along the z-axis, what the other one did. And so you can combine all of these views together to create something like this, where now you have an isotropic resolution in X, Y, and Z. So this is just like a scan here. It's not changing over time. I'm just scanning through my sample here, like in Z, that's confined to this tube, and here, like along like this axis.
So you probably borrow algorithms from computed tomography and
So
CT scans and stuff?
so here to kind of, there's a couple of things. So to kind of reconstruct the 3D website, we have bead in different colors,
Okay.
and that forms kind of like a 3D puzzle. So we can reassemble the bead channel, and then we apply the same transformation to the microtubule.
Yeah, tomography, the images are projections, right? Here they're slices.
Sorry, say that
In
again.
tomography, the images are projections, right? Here they're slices.
See the
It's
slides.
not the same, yeah, thank you.
And we also use a lot of deconvolution to try to get really an isotropic point spread function. It's going to be important because really what we want to measure is the anisotropic of the Fluorescence signal. We want to try to detect in which orientation of all those rods are pointing. So if you have some aberration and like all your rods look like, all your points look like rods, it's going to really impact the measurement. Anyway, so so this is kind of like a very high data throughput experiment because 1 hour of imaging gives like 1 terabyte of images. So we had to kind of think about ways to kind of decrease the size and kind of extract like the dynamic of the system without looking at terabytes of data. And actually, topological defects are a great way to do that. So what we've done to get the defects in 3D, we first map the orientation field in 3D. And so you can see that you have kind of domains that are like uniform in orientation, but if you look further away, they have not correlated. So between those domains, that's where you have the high nematic distortions. So taking the gradient of the director field tells you where you have the highest distortions. And then when you look how the director field winds in this region, you can definitely confirm that those are topological defects, so singularities in the orientation. And so now I'm showing you like a 3D reconstruction of the topological defects in this material. They form these networks of lines that are spanning the entire system. And you see some of these like isolated loops like here. Right, so those are like the defects that you see in 3D. Those are like linear, curvilinear structures. And like people have seen this thing in like passive liquid crystals. Right? What's unique here is that they're very long-lived. And actually, you nucleate those defects as well. So you can really study them for a very long time. And you're able to kind of really, because we have the orientation field everywhere, we can measure the winding around the defects. Right, so that's quite different from like what I showed you earlier with the 2D, where now you have point defects, plus half and minus half point defects. Okay, so when we look at the dynamics of those defects, we see nucleation and annihilation. We see isolated loops that can nucleate from a uniform background, and isolated loops that can collapse on themselves and disappear without leaving any defects behind. Right? So that tells us that this defects loop have to be neutral. There is no defect at this.
What's the scale here?
I think those loops are probably 20 microns in diameter. And the microtubules are micron size.
Sorry, can you say also something about the time scale of this experiment?
Yeah, so here we're able to take a picture every, I think, 20 seconds.
And the embryos are fine?
Sorry,
The embryos are fine?
the embryo.
They're okay. Are they okay?
Okay.
You mean for how long can we measure or something?
Yeah, no, I'm just wondering if killing your embryos.
Oh, no, it doesn't. That's the point with like light chain microscopy compared to regular, because you.
These are
really-
actual embryos?
No, it's not an embryo.
No.
That's like so first of all, that's not an embryo. So it's
in
very
vitro,
common.
right? Yeah.
But you can. That's the advantage of light chain microscope compared to like wide field or confocal, is that
Efficient.
if that's my sample and that's my light sheet, like, and I look from here, I really just like illuminate what I'm looking at. So everything that's like not in the light sheet, I don't expose, so like I don't damage it.
Yeah.
While if you were to use, a wide field, like you can focal, like you still look at just this, you have to scan, but you still illuminate a little bit like everything else.
So presumably the next step for this beautiful stuff is to do a biaxial nematic, some active biaxial nematic, because there the defects have non-commutative,
Yes.
and they get entangled and they get strings. Is there any hope for doing that?
Okay, so
Surely biology has provided a biaxial nematic can be run on ATP.
so what we really hope to do is instead of using colloidal particles that already exist from in biology. So here we use elongated viruses that are very well defined size, but like
That's FD.
we use it. We hope to use maybe DNA organic. So you can create your own particle, whatever shape you want, whatever orientation you want. And so this is why we did some work like a few years ago where I was able to build like cholesterol liquid crystals out of DNA organic. What's a little bit more tricky here is that because we need to dope those colloidal liquid crystals with the microtubules, you actually lose the cholesteric structure because the microtubules are like the bundles basically are much longer than the pitch, the cholesteric pitch. And because those two, cholesteric and nematic are so close in terms of like energy, so we don't see any cholesteric pitch here. So I think that's one of the drawbacks of having these microtubules that drive, like the background is that you have those long bundles, and so that still prevents the type of, like local kind of order that you could. But we have down the line, I'm sure we're going to be able to like do some more complex.
Well, biaxial nematics would be great to do that in active form.
One more clarification
Yes.
question. I'm sorry if I missed it, but you mentioned the timescale of doing one scan, but how is it compared to the timescale of like one microtubule slide over its whole volume?
Okay, yeah, so that's a good question. So like we can tune that experimentally. So if you decrease the amount of motor or you decrease the ATP, it moves slower. Okay, so we're able to do the whole scan, I think, in 20 seconds. And that's still like good enough to be able to kind of track the nucleation of those defects. But we have to slow it down really, so to be able to, because you don't want to do a scan and that your material kind of like moves as you do the scan. You want to scan more faster than the rate at which it's deformed. Right? So we have to slow it down a bit.
Can you control the length of the microtubules?
Yes, we can do that. Fantastic. And that leads to a lot of communication actually. Okay, so we can, because we have the director field everywhere, we can look at how it winds around the defects. So this is a defect loop here, and you see this cross section, so you recognize kind of those profiles that we had in 2D as well, the minus half wedge profile and the plus half wedge profile. What's unique here is that now those things are connected. So it means in 3D, you can continuously deform this minus half into the plus half, which you cannot do in 2D, right? In 2 dimension, those point defects are topologically distinct, so it means that you cannot continuously transform the minus half into a plus half. Now, because you're in 3D, you can do that, and that's because you can twist, right? So if I look at the rod here, just below my defects, I can continuously rotate them out of plane, so twist them and transform. So now they're sticking out of plane, and I can transform like this minus half into a plus half. And because we have the director field everywhere, we can also measure the bend, plane, twist. And we did see that between this plus half and minus half wedge profile, we see some twists. All right, so that was done. Work that was done with Denver, that like some of you might know because he was doing a postdoc here a few years ago. And so what we've done then was trying to classify the types of defects that we see, the type of loops. So this is a loop I described earlier, right? If you look around, you go from like this minus half wedge profile, you twist into a plus half, and then you twist again into the minus half. Okay. And so that's kind of what we were expecting from this bend twist instability, right? That's the types of defects that will grow. But what was surprising is that when we look at statistics of, like, when we looked at the type of loops, we actually seen a lot of pure twist loops as well, which are also, you know, like neutral loops. Except that now, if I take any cross section on this loop, there is always some twists. So like the rods would never, all of them would never kind of lay in a plane of cross section.
Neutral means the whole thing is not a topological defect. Far away, you don't see this.
Yes, yes, so it's like it can collapse in the third zero.
So just a corollary, I guess, is that there's no conserved quantity that you can construct with a loop integral.
Yes, yes.
Nevertheless.
So actually, most of the loops that we've seen were purely twist loops. Okay, and that's super weird because we were expecting to see bend twist loops from wedge twist loop from the bend stability. So, and also like it's not that like you have wedge nucleation of only wedge twist loop that then turn into pure twist loop. When loop nucleate usually stay the same. Okay, so it means that we actually were seeing a lot of nucleation of pure twist loop. Yes.
In just regular passive liquid crystal elasticity, can you calculate the energies of each of these, and is one bigger than the other?
That's a good question. So you can, and that depends on kind of the different elasticities for like the different modes. And so
You know that for your system actually?
So
ratio elastic constants.
some of them have been measured for like this corridor system, but not all of them. I think also because it's cholesteric and not in nematic crystals, the twist is much lower. So, okay, we've done simulations of this as well, where you have a constant nematic elasticity, and that doesn't account for this difference between purely twist and wedge twist. So even with like 3D active nematics where you have the same cost for bends, bend, and twist, you still see mostly the nucleation of pure twist
In the simulation.
in simulations.
And that is not explainable by just some simple energy.
No. Instead, so when we look at how those loops nucleate, it seems that it's because you have you're in this kind of chaotically moving liquid crystal, and so you can have situation where you have twist that builds up in different, like you have flows in different planes and twist that builds up locally. And so here as the twist is increasing, you basically again want to relax the distortion by nucleating a defect that then grows over time as those planes get more and more twisted. Okay, so that's like a different mechanism here. It's more like the byproduct, let's say, of like all those like flows, band-driven flows that happen everywhere in different planes, which leads to the nucleation of this pure twist loops.
But is the plane of these defects correlated with the plane of the plates? I mean, this is still
No.
narrow.
So,
It's
so now
it's
it's
not narrow in one
no.
direction.
Now you have like this
Or is it
tube.
more like a cube.
Yeah, you see you have this tube now that's like 800 microns and like very long as well.
But the orientation is not correlated with the 6 planes of the cube, perhaps?
So it was initially, but like
Let's forget.
Yeah.
Thank you.
Okay, so any questions here about the defects? And so those charge neutral disconnection loops are basically the primary topological excitation of this 3D active maps. All right, let's take a step back now and think about the instability of it. So this extensile flow, right, drive the in-plane by instability. What we've done next was to try to tune the molecular composition of this material, right? So you have motors, microtubules, ATP. What we've done was to reduce the ATP concentration, so the fuel used by the motors to kind of step. And right, so when we do that, we still see something that's unstable, but it's a very different instability. And so you see those patches that are getting like kind of blurry here. You see have a finite wavelength. So what's happening here is that it's buckling out of plane of the.
What's that strange white dot that's dancing across the left side of
This thing.
no, there it is.
Yeah, I don't know.
Little bacteria,
That's-
maybe a little
maybe
bacteria.
bacteria could It's a very slow time scale.
No, I know, I agree, it's not the most important thing.
It's blurry because the objective has certain depths of field, so as when you go outside of that, it just looks blurry. But if you put this inside a confocal, you can clearly confirm that the system is like buckling this time along the z direction. Okay. So again, it's still a very now we're back to this thin sample, 100 microns. So to kind of quantify this transition from in to out of plane, we use image analysis to kind of detect, segment the image, segment the sections that are like blurry, and then we look at the fraction of the image that's blurry versus like the total area of the fraction. So when this blurriness parameter B is equal to 0, it means that you're always in plane. When it's greater than 0, it means that you have some out of plane features. And what we were thinking, the hypothesis back then was that this is akin to like a liquid to solid transition. In the in-plane instability, we already shown that this is what's happening in liquid crystal. You have the strong confinement along Z that basically slow down the fluctuation and it flows in plane. Now this is more reminiscent of what's happening with like elastic sheets. And the other kind of clue we had why it could be due to different elasticity in these materials is that we know that in this type of network, when you start to decrease the ATP concentration, so the fuel, the molecular motors basically cross-link the microtubules. So instead of walking, they just basically bind and cross-link everywhere. Right? So basically, if you have a system with no ATP, you just have this like cross-link viscoelastic gel. So decreasing the ATP has two effects: is decreasing the total kind of stress generated, but it also increases the cross-linking density. And so we quantify all those things. We change the motor concentration, the ATP, we change the amount of cross-linkers, and every time we see something that's consistent with this liquid and solid transition. So decreasing the ATP, increasing the ATP here, you go from an out of plane to an in-plane stability. If now instead I take this in-plane stability and I start adding passive cross-linkers, I go to something that buckles more out of plane. So increasing the elasticity again, the cross-linker increases the elasticity of the network. If now I do the same thing with motors, we have this interesting re-entrant transition. So at low motor concentration, you have the out of plane buckling. If I add enough motors, then I fluidize my system at in-plane stability. But then if I keep adding motors and there's not enough ATP, then it's basically like adding cross-linkers again. And so we go back into this out of plane bucket.
Can you measure a shear modulus of the gel?
So it's possible to measure the elasticity, elastic and the G and G prime basically using rheology. And Zvonimir did that with Dan Blair, and they got and so actually we're going to use some of their like measurement to kind of estimate the stresses generated in the system. But yeah, so like we quantify all of these things, and then we kind of talk again with Aparna, and she was like, okay, let's add elasticity to to the model we used before for the 3D actin nematics. Okay, so now you have an extra term in your free energy that basically accounts for like elastic deformations. And so when you do that, you can build this phase diagram, activity versus bending motors. And you observe two distinct instabilities. The out of plane stability that's always there. So what even at very small activity, you're always going to buck quality. And you still see the in-plane stability, but this time you need a critical active stress that's greater than the elastic motors to kind of trigger the in-plane stability. And then how we build this phase diagram here is to look at which modes grow the fastest. And so in this like orange area here, that's where the in-plane modes grow the fastest. In this intermediary region here, the out-of-plane modes grow faster, but you can still have an in-plane stability. And below this line of slope 1, that's where you have no more in-plane stability and only the out-plane. Yes?
There's always an instability.
So in this one, there's always an out-of-plane stability.
This,
Any small finite activity will trigger.
because, you know, the biological structures that are most like what we're talking about, spindles, are stable. So what's different? Why doesn't, what in your model doesn't look like a spindles?
That's a good question. So now maybe there's something more complex with spindles. It's probably like maybe other proteins that will stabilize this thing.
Spindles probably evolved over 2 billion years to avoid this.
So okay, so when I say it's always,
That's
okay.
cheating.
You can hear very slow instability too. Could be a question of time scale. You know, like how long is a spindle stable for?
The day.
Infinite today?
Long time.
I know. Okay. Okay. So that doesn't work.
We can talk about this. I find this very puzzling. We should talk about this later.
So yeah, but that's a good question.
One more good.
Yes.
So where is the state of system in your previous 3D experiments in this one?
Okay, so we're like in this region here.
Oh,
This
rather,
region, yeah. Actually, yeah, very low proximity. Okay, so what my student Najma did is she did like tons of experiments. So here, each data point is one experiment that's been replicated. So it's really like massive amount of work to try to build these experimental phase diagrams here. And also massive amount of work to try to do that reproducibly. It's very hard with protein to kind of make sure that from one day to the next you get the same results. So what she's done is to prepare a extremely large volume of this and then flash freeze everything. Then every day she was able to kind of, you know, like change a little bit the experimental conditions to build these 4 phase diagrams here, where we change basically all the kind of molecular parameters we could imagine, the length of the microtubules, the amount of motors, ATP. And then what we've done is to kind of use enzyme kinetics to try to go from kind of what's happening at the molecular composition to the material properties. So that allows us to really then compare the theoretical like phase boundaries with the experimental phase boundaries and fit the theory to that. And now because we can do this fit, it's not perfect, right? But it still captures the main features. We're able to try to, measure all the parameters in this active gel theory that the parameter develops. So the activity and the shear modulus. Knowing what so actually in the theory, by fitting, we get the ratio of all those things, but because it's been one of them, the shear modulus was measured in a certain kind of concentration of everything by that in volumeters, and we're able to use that to kind of really estimate all those parameters. And so now what's good is that, you know, like what we've done so far is take those blocks, build something, now we can do the reverse, right? Because if you tell me, okay, I want an active material that has a certain elasticity or the ability to generate that amount of Pascals, then we can look at this kind of multi-scale, like maps here and really tell you how much motors we need, how much processing curves, what's ATP constitution. Okay, so the conclusion is that by we can infer the mechanical properties of these materials by basically looking at what's the dominant mode, the most unstable mode inside the system. You have a little bit more time, or
I would say another 5 minutes since we started late.
Yeah, I think so. Other-
So let me briefly talk about this last part. But most of the kind of active materials that are other experimentally are like driven by molecular motors. But cells have other mechanisms to generate forces. So for example, here, the way this cell is moving forward, if you zoom in here, you have this branch network of actin that's actively polymerizing against the leading edge and that push the cell forward. Right, so that's driven by the non-equilibrium polymerization of actin. What's happening is that it's also an ATP-driven process that basically biases the polymerization and depolymerization rates. So now you have mostly polymerization of actin on one end and depolymerization on the other. And so what we try to do here is use this to kind of design a new class of polymerization-based active materials that doesn't require any molecular motors. And so we don't need to make it from scratch. People have, biology knows how to do that already. So this is a mammalian cell here that's infested with a parasite, Listeria. This parasite is actually covered with a protein that nucleates a branch actin network. And so now, because you only have polymerization here, the Listeria is hijacking the actin of the host cell to propel itself. So if you take this and you put it just in water, it's not going to move. It requires the actin of the host cell. And so,
And ATP as well.
and ATP, yeah. So ATP, yes, ATP. So you can have polymerization without ATP. The ATP is really what's biased the ends on both ends.
And the polymerized tail behind this propelled Listeria is gradually dissolving with time.
Yeah, so it's depolymerizing here
Okay.
and it's polymerizing. So that's actually pretty cool. So 20 years ago, a few labs were able to kind of study this and reproduce this in vitro, where instead of using Listeria, you take like polystyrene beads that you coat with an actin nucleator, and instead of using a mammalian cell, you can either use extract or you can purify all the proteins. And that was hard work because it requires a lot of different types of proteins, of actin-associated proteins. You need the one that nucleates on the bead. You need the one that stabilizes the tail, because you don't want to have growth of those like polymers here. You just want to have the polymerization here. And you want also proteins that can depolymerize, that can replenish the pool of monomers that can then be used on the bead. And interestingly, that's really the rate limiting step here. It's a depolymerization. And so that's why those beads were like kind of like they're very slow. They're much slower than what's happening in vitro, in vivo, so the Listeria. And even if you look at kind of in vitro polymerization of actin, it's very slow compared to what's happening inside cells. That's mostly because we back then didn't know how to enhance this polymerization rate. And so we've been working with Bruce Good, who's been pioneering kind of like how we can use cocktails of different actin-associated proteins that to enhance this depolymerization rate, to kind of chop basically the actin and then increase kind of the turnover from filament to monomers. And so using that, we've been really able to kind of get now really like fast moving.
This is a short summary that when you can't depolymerize rapidly, you have to slow down because you use up the actin.
Yes,
the
you have to.
depolymerized actin.
Yeah, you basically deplete your actin. You have to wait that you have another monomer that can diffuse around.
So Listeria can also, as you probably know, bore between neighboring cell walls and infect new cells. No one's been able to do that with colloids.
Interesting. Yeah, I don't know. Not yet. From what I know.
Good. Don't do it.
Anyway, so this is just like a quick summary of what people have done with this release, and we're focusing on like a single bead. to kind of understand this polymerization-based motility, or to do some biochemistry and kind of identify what proteins like regulate this process. What we want to do instead here is try to create an active material, so active colloids that will kind of bump into each other and see what type of pattern emerge. And so we've done a lot of protein purification, a little bit of mixing, and then we're able to kind of reproduce these assays in the lab. So here you see, those are, I think, 3 micron or 1.5 maybe micron beads that are moving around. And you see them here in phase contrast. So this is a bead, and you see the tail of actin here that's behind. So a lot of time scale here is very slow. It's probably, you probably have a minute or two between frames here. But it's still, faster thanks to this SRV2 vaporization process. The issue is that those don't interact. So we try to increase the density, we never see any flux. We were hoping to see some type of polar motion. So what we try instead is next is to increase the size of the beads. And then when we did that, we got some very nice flux. There's a microscope. So those are 6 micron beads. It's still confined between 2 plates, so it's in this quasi 2D geometry, and you see a lot of very complex things. And so the question we had then is like, why do they flock at all? Why do they move this way? Is it like some of this? Is it like density dependent? Does it is it like a motility in this phase separation where they have a slow kind of rotational diffusion, and because it just collides, they kind of stabilize clusters like this? We don't think that's what's happening. So what we've done first is to kind of try to change the height of the chamber, and that basically controls the steric interaction. If the chamber is too high, then they can tap on top of each other. That's why we didn't see flux in the first place. And so we use the same bead as before, 3 micron beads, and now we kind of like squeeze the plate. And when you do that, you kind of recover flux, right?
I mean, this is very modest flocking. You get 3
Yes.
at a school or something.
Yes, but it's still happening, right? It's still kind of very stuck to each other. And we did the reverse too. When it's the one that were flocking the 6 micron beads, when you kind of increase the gap, then they stop moving.
Why
And when
are the active tails shorter on the right?
That's a good question, but I'm not sure.
I mean, there's less material, right? Like,
Yeah, but everything's scaled, right? You have less beads too. Actually, no, maybe I think we increase the number of beads here. Kind of, make sure that descendants.
by the
So it could
volume.
be, yes. So it could be that. But steric interactions are not enough, because if we go to very small beads, 1.5 micron bead, and we squeeze them, we see that indeed they interact, they align, but they don't flock. Right? You see those two, they come together, but then they really don't stay with each other. So it's quite different from what we had in the 6 micron bead that collide and then keep going, right? And also when we look at the shape of those flocks, that's quite unique. It's not like isotropic. You see those things that line up like this. So our hypothesis now is that you have the actin concentration is actually important. You have something, they interact because they kind of locally kind of consume the same fuel, actin monomers. And so imagine you have one bead, it consumes actin, so you create a local kind of actin gradient like this. Now, if you have 2 beads, because they consume the same protein, you're going to see less actin or less ARP, I don't know, between them compared to the left, right? And so because the force they feel depend on the actin concentration, we're going to see a larger force on the side and center that's going to create an effective attraction between the 2 beads. Okay. So that's kind of what we're trying to test now. If this is true, there is one prediction is that if we were to change the actin polymerization rate, we should be able to change the flocking. And so we did that. We changed the amount of actin nucleator you have on the bead. And so that's the same movie as before. When you have 100% coverage of the bead, if now you reduce it to 1%, same bead size, you lose all the flux. Okay, so those guys still move, but now, and they still kind of align to each other, but then they don't kind of stay. Okay, so the conclusion here is that actin provide base can interact sterically, generate polar flux. you can change the interaction by, like decreasing the bit size, so which also decrease the total, like the actin consumption, or by changing the coding, by changing the actin consumption rates. And now we're trying to kind of use reaction diffusion, like modeling to try to better understand what's happening here. All right, so I'm going to just quickly conclude some over time. I showed you that we can use purified proteins to kind of create a lot of different types of active materials. We looked at active liquid crystals made of microtubules and motors, active viscoelastic gels. We've done that in 2D, in 3D. Now what I had at the end was trying to use like motor-free materials where it's not driven by molecular motors, but instead by like the out of equilibrium turnover of the filaments. And so with that, I'm going to skip some of this conclusion slide and thank our funding from NSF and MERSEC, and thank you, Marianne, for like correcting our data and doing all the work.
Great, thank you for the talk. So there's been a lot of questions, but I guess we haven't been some if there's any other questions either from the Zoom audience or from the.
Question. So I think it's very amazing that you try to start from enzyme kinetics and sort of to explain your data for the instabilities. So I wonder, like, does that requires you modeling how the material properties also depend on its ATP levels and as well as how active stress will be and all that. So because
yes, we did that. So basically, we say that the activity depends on the elongation rate, right, which depend on the ATP hydrolysis rate.
Right. But at the meantime, I think the material properties themselves are.
Oh, the material properties, yeah.
So,
exactly. So we said that so there's been some studies that look at passive materials, how the like elasticity depend on cross-linking density. So we use those studies to kind of estimate really basically the cross-linking density with the material properties. And then for the cross-linker, we say it's the sum of the passive cross-linker PSC1 and all the molecular motors that are not bound to ATP. So that's where the chemical enzyme comes.
I see. Right. Maybe this is a little bit details like related to active stress in there. Usually, in active people put like kind of a activity coefficient in front of that, and like we have to estimate the magnitude of that.
So we that's what we did. We didn't have to estimate it. We fitted it. Right. So that's the efficiency you're talking about, right? Yes, we fitted that. But we have one fitting parameter in front of the activity, and that's how we're able to kind of get estimate like this of this activity. Now, like basically we say that like to be able to, the bottom line is that to be able to move like a liquid here, you need to generate stresses that are greater than the elasticity. And so therefore, because the elastic modulus measured for the system were like around like 50, 60 pascals, then that's how we get the estimate from this. Yeah, I skipped a lot of stuff.
Yeah, but this is very cool. I think this is probably years of work to just skip there, but it's very amazing.
Right. Other questions? Oh, I guess we've century, so let's think about that. Is there a coming? We'll stay tuned for next year. Maybe.